Hello, everyone, and welcome to today's webinar. I'm Iris Klobatsky, Associate Science Editor for The Scientist, and I will be moderating our discussion. Today, Anisha Munoz-Bull, Jens Christian Schwamborn, and Juan Eduardo Rodríguez-Gatica will discuss using PSC-derived midbrain-striatal assembloids and a 3D light sheet microscopy workflow, highlighting its value not only to elucidate Parkinson's disease-related pathomechanisms, but also to accelerate the discovery of potential therapeutics. Before we begin, I'd like to thank Miltenyi Biotec for sponsoring today's webinar. And with that, let me introduce our speakers. Anisha Munoz-Bull received her doctoral degree in biology from the University of Cologne, Germany in 2023, with a focus on neurodegenerative disorders. After her studies, she joined Miltenyi Biotec and currently is the global product manager for the neuroscience portfolio. Closely working together with research and development, Anisha focuses on innovative solutions that support neuroscience research from basic to translational stages. Jens Christian Schwamborn is the head of the Developmental and Cellular Biology Group at the Luxembourg Center for Systems Biomedicine, as well as a professor at the University of Luxembourg. He obtained his PhD in biology from the University of Munster and subsequently worked as a postdoctoral researcher at the Institute for Molecular Biotechnology in Vienna. He was previously the co-founder and chief scientific officer of Brain-Engineering Technologies in 2019, and he co-founded Therapeutics, where he currently acts as CEO. His current research focuses on neurobiology, stem cells, and Parkinson's disease. In particular, he is interested in using human-induced pluripotent stem cells for the development of brain organoid and assembloid models, which are used for in vitro disease modeling. Juan Eduardo Rodríguez-Gatica is a postdoctoral researcher in the Functional Neuroconnectomics Group of Martin Schwartz at the Institute of Experimental Epileptology and Cognition Research of the University of Bonn Medical Center. Completed his PhD in the Biophysical Chemistry Group led by Ulrich Kubacek at the University of Bonn. His current research involves circuit analysis of neural connectivity in 3D image datasets acquired by combining expansion and light sheet fluorescence microscopy. Okay, let's make sure our slides are up and running.
Thanks, Diana, for your kind introduction. First of all, I want to send a warm welcome to all of you. Thank you for joining us at today's webinar. Here is my disclaimer. Before we start, I will present you today's agenda. I will kick off the session with a brief introduction into the integrated workflows for PSC-derived cerebral organoids that we offer at Miltenyi Biotec. Then I will hand it over to our first speaker, Professor Schwamborn, who will talk about the use of PSC-derived midbrain organoids and assembloids to model Parkinson's disease. Afterwards, Dr. Rodríguez-Gatica will explain what is light sheet microscopy and how can we use the ultramicroscope blaze to image organoids and assembloids. Finally, we will open the floor for a Q&A session to address the questions you may have. Miltenyi Biotec was founded 35 years ago in Germany. Currently, we are more than 4,900 employees, with more than 20% of us dedicated to research and development. This fact highlights our strong commitment to develop innovative solutions to researchers and clinicians. With more than 19,000 products comprising instruments, reagents, and consumables, we are able to offer integrated workflows for a vast range of applications. At Miltenyi Biotec, we offer integrated workflows that support your PSC-derived organoid research. First, we have a reprogramming workflow that uses our STEMMAX iPS mRNA reprogramming kit, which has been designed for rapid and highly efficient reprogramming of human fibroblasts into iPSCs. Next, our PSC maintenance and cryopreservation workflow with our PSC brew media is designed to allow flexible feeding schedules without compromising pluripotency of the cells. Moreover, our broad cell culture portfolio allows you to flexibly select the perfect solutions for your PSC differentiation needs. Once you have your organoid or assembloid, to analyze them in one hand, the cellular analysis workflow starts with the gentle dissociation of the organoid. To do so, we combine the mechanical dissociation with our gentle max dissociators together with enzymatic dissociation provided by the neural tissue dissociation kit. This mechanical and enzymatic dissociation combination ensures maximal cell viability. The resultant single-cell suspension is ready for downstream applications such as flow cytometry, genomic analysis, or cell culture. On the other hand, the 3D imaging workflow uses light sheet microscopy to allow you to study the complex neural networks within your organoid. The combination of these two workflows allows you to gain an in-depth understanding of the organoid model, from the analysis of single cellular components to the visualization of the neural architecture and the neural interactions. But for today's webinar, we will focus on the application of the 3D imaging workflow analysis of organoids and assembloids. To visualize the neural architecture of the organoid, we need to follow a series of steps to prepare the sample for the light sheet microscopy. The first step is the staining of the organoid. We know that optimizing a staining takes a lot of time and resources and not always means you will have good results. To make it easier for you, we have validated our antibodies for 3D immunofluorescence and optimized them for deep tissue penetration. This will help you reduce the time and cost you invest during the optimization phase. In addition, our antibodies are conjugated to our fluorochrome dyes, which will lead you to obtain faster results, since you don't need to incubate with a secondary antibody. Also, our antibodies are recombinant, which will enable you to generate reproducible data. Here at the right, you can appreciate the differences between two beta-3 tubulin antibodies. At the left is a staining with a non-validated, non-optimized antibody, and at the right is our beta-3 tubulin antibody. As you can appreciate, already validated antibodies will allow you to visualize the different structures within the organoid. You can browse through our website to find out more 3D immunofluorescence validated antibodies that can fit your experimental needs. The clearing step is crucial if we want to visualize the 3D structure using light sheet microscopy. Our max clearing kit is organic solvent-based and provides you with fast and efficient clearing results. It is non-toxic, which makes it safer than other clearing methods. In addition, we have optimized clearing protocols for different tissues that you can find online on our website. Here you have an example of the steps and timing for the preparation of an organoid for 3D imaging using our reagents. The last step of the workflow is the imaging using the ultramicroscope Blaze, which is a fully automated microscope, meaning the microscope will image your samples automatically, but you can do other experiments in the meantime. The microscope is easy to handle and has cutting-edge optics that allow you to obtain high-resolution images from whole mouse to small organoids. In this video, you can see one of our last releases for high-throughput 3D imaging, the UltraMount 48, which allows you to place up to 48 organoids and image them in one run. For this video, our R&D team generated cerebral organoids and differentiated them for 30 days. The organoids have been stained with the proliferation marker Ki67, the neuronal marker beta-3 tubulin, and the neural stem cell markers PAX6 and SOX2. In addition, all the organoids have been cleared with our max clearing kit, following the protocols we have available in our website. And with this, I will now hand it over to our first invited speaker, Professor Schwamborn from the University of Luxembourg.
Hello, my name is Jens Schwamborn, and I'm going to talk about modeling Parkinson's disease in patient-specific midbrain organoids and actually also midbrain striatum assembloids. Before going into the actual presentation, here is a disclosure. Some of the technologies, of the approaches, of the molecules that I'm going to present in this presentation are licensed to the company Therapeutics that I co-founded a while ago. So Parkinson's disease is an increasing problem for our societies because particularly in the Western world, but also in other parts of the world, societies are significantly aging. So we are all becoming older and older, and Parkinson's is age associated. You just see here a curve from a paper that has been published already a couple of years ago. And actually the current numbers are already higher than what is projected here. We currently estimate probably having about 10 million Parkinson patients, and numbers are strongly increasing. So it becomes a higher burden for society. What is Parkinson's? Parkinson's is a neurodegenerative disorder. I think all of you already have seen patients with this disease, particularly identified on the motor symptoms. These are tremor, bradykinesia, rigidity, many others. But there's also the non-motor symptoms that are pretty diverse. Different patients suffer from different of those non-motor symptoms. They include sleep disturbances, hyposmia, anxiety, and many other aspects. On the histopathological levels, in healthy conditions, dopaminergic neurons, so neurons that produce the neurotransmitter dopamine, have their cell bodies in the substantia nigra of the midbrain, and there they project to the striatum where they then secrete dopamine. And that is important, particularly for muscle movement control. While in the disease situation, these dopaminergic neurons degenerate. As a consequence, levels of dopamine in the striatum are much lower, and it comes to the symptoms that I've just mentioned before. And since the substantia nigra has its color, its dark color from neuromelanin, we can see that under normal conditions, this dark staining is present, but it's absent in the Parkinson conditions just because these neurons are degenerating. On the other hand, the disease goes hand in hand with the appearance of protein aggregates consisting of the protein alpha-synuclein, and this is called Lewy pathology. That can be Lewy bodies being intracellular, actually in the neurons, or in the neurites, and then are called Lewy neurites. Parkinson's disease has risk factors as well as inverse risk factors. Risk factors are genetic factors. There's about 10% of all cases are clearly monogenetic, but there's also a larger population of idiopathic, which can have a complex genetic, have environmental contributions, or just an unknown cause. Aging is probably the major risk factor, so the older somebody gets, the more likely it is to suffer from Parkinson's. And I already mentioned that environmental factors, particularly, for example, pesticides, but also air pollution, strongly contribute to the risk to suffer from Parkinson's. There's also inverse risk factors, including female gender, certain lifestyle factors, obviously a healthy lifestyle with sports, a complete diet, are helpful, but also unexpected things like caffeine consume and smoking seem to be inverse risk factors. And then there's genetic factors that are also protective. So our approach to model Parkinson's disease in the lab is the usage of human midbrain organoids. So everything that we are doing is starting with patient samples. Typically it's going to be skin biopsies, but it could be also, for example, blood or even cells that you can find in urine. So these cells are then reprogrammed with the Yamanaka factors into induced pluripotent stem cells. These are stem cells that are very similar in all their characteristics to embryonic stem cells. We further differentiate them into neuroepithelial stem cells that are already pre-patterned for midbrain-hindbrain identity, and then we differentiate those into the actual midbrain organoids. And the reasons for doing this intermediate step is by restricting the differentiation potential. The midbrain organoids that we get out of this process are actually highly reproducible. Just in a nutshell, what are midbrain organoids? These are 2 to 4 millimeter size, 3-dimensional structures, have midbrain identity, so they express transcription factors like FOXA2 or LMX1 that identify these as midbrain cells. They have high levels of dopaminergic neurons. You see here the TH staining for dopaminergic neurons, but you also can see dopamine in green here itself being produced within those cells. The midbrain organoids produce neuromelanin, which is quite interesting because this is a feature that you would, for example, not find in rodents. And that's actually a feature of maturation, already indicating us that although these organoids are probably young because they are derived from embryonic cells, they do have features of maturity. Midbrain organoids show a tissue organization, so we have a layering of cells, and we not only have neurons, but we also have glial cells and other cell types, and the neurons in there show electrophysiological activity. A subset of those neurons actually also show pacemaking activity, firing with a frequency of about 6 Hz. We used a single-cell RNA sequencing approach to get a better understanding of the actual cellular composition of those midbrain organoids, and that was done together with people in Luxembourg, like Alex Skupin's lab, as well as the Riken Institute in Yokohama. And what we can see is, if we compare midbrain organoid of a day of 35 and 70 days, is that the cellular complexity is increasing. So we get more cell types when we continue to differentiate them. And we not only find dopaminergic neurons, but also other neuronal cell types. Just as an example, we do see, for example, glutamatergic, GABAergic, or serotonergic neurons. So we have really a huge degree of complexity. Now, having these midbrain organoids in hand, we obviously want to use them for in vitro modeling of Parkinson's. And what we did in order to do that is looking for the three main features that Parkinson probably has. This is a loss of dopaminergic neurons, the appearance of alpha-synuclein aggregates, and neuroinflammation. So let's first look at the loss of dopaminergic neurons. Here we differentiated induced pluripotent stem cells with a mutation in the LRRK2 gene, a mutation that leads to PD, either in 2D or in 3D, in dopaminergic neurons or midbrain organoids, and then simply quantified the amount of dopaminergic neurons. And strikingly, in a 2D culture, we would not see this loss of dopaminergic neurons that we see in a patient. While in the midbrain organoid, we can very clearly see this phenotype. You can see here in the red column, the PD situation has way less dopaminergic neurons than the healthy control. Further, we do see the appearance of protein aggregates. This is here a setting where we have healthy midbrain organoids, so from an individual that does not suffer from Parkinson's disease, as well as from a Parkinson patient with a triplication in the alpha-synuclein gene. And you can see in the healthy situation, there's a bit of synuclein in the green staining, but we do not have the phosphorylated version, the P129 version of alpha-synuclein, which is a hallmark of the pathology. While in the disease situation, we have way more alpha-synuclein, the green staining is way more intense, but we also particularly find these aggregated P129 positive structures. And if you zoom in there in more detail, you can even see that they form quite diverse morphologies, pretty similar actually to what we see in the patient brain. And to highlight this in a bit more detail, we compared the different morphologies of these aggregates in the organoid to what we actually see in the patient brain and post-mortem samples. And again, we do find that many of the structures we can see in the midbrain organoids are similar to what we see in the patient brain, convincing us that we really can here recapitulate pathology to a good extent. And importantly, in this setting here, we do not trigger in any way. This is really endogenous just because the organoid comes from a patient. There's no fiber treatment, no toxin treatment. It's really just endogenous. And then the third characteristics is the recapitulation of neuroinflammation. We can look, for example, for the levels of pro-inflammatory cytokines. And what we would see is that in a midbrain organoid from a patient with a triplication alpha-synuclein, we find significantly higher levels of interleukin-1, while in a patient with a LRRK2 mutation, we would find higher levels of TNF-alpha. It is interesting to see that in both cases, we would have, in general, pro-inflammatory conditions, but the primary cytokines that drive these conditions seem to be different depending on the mutation. Importantly, we can not only recapitulate the disease, but we can also use strategies to develop therapeutics. And I just show you two studies here that we have published in the last years. One on the left side is with patients with a mutation in the PINK1 mutation, where we get again midbrain organoids, and you can see that in the diagram, the patients, this is this red color, have way less dopaminergic neurons than the healthy controls. But then upon treatment with a drug, in this case is a cyclodextrin, we can rescue the phenotype to a good extent, meaning we can increase the amount of dopaminergic neurons. And that drug here works in a way that autophagy is turned on. While in the second study here on the right side, this is from induced pluripotent stem cells with a mutation in DJ1, which is also associated to Parkinson's disease, which is a splice site mutation. We can rescue by treatment with splicing read-through compounds. And again, you can see that the patient situation or the PD mutation situation has way less dopaminergic neurons than the healthy control, while upon treatment, we can rescue the amount of dopaminergic neurons significantly. So this is a model that allows us not only to describe the disease to a good extent, recapitulate hallmarks, but also to experimentally test therapeutic approaches. However, you remember from the introduction, Parkinson is a complex disorder where the dopaminergic neurons have their cell bodies in the midbrain, and this is probably what we have recapitulated so far in the organoids, but they project their axons actually to the striatum, and this is something in the model that I've shown you so far we have not recapitulated. So we set out to come up with a more advanced approach where we take midbrain organoids and actually fuse them to striatal organoids, so organoids representing the striatum, and build midbrain striatum assembloids, where this projection from the midbrain to the striatum would be recapitulated. And yeah, we have developed these midbrain striatum assembloids, and in a first attempt, obviously, we want to characterize them. So we look for the expression of certain markers for the different regions of the brain. Like, for example, as I've told you before, the midbrain would express FOXA2, you can see that here in the red staining, while the striatum would express CTIP2, which is the yellow staining here. We find ASCL1, again, it's a striatum. We find choline being expressed in the midbrain. And obviously, the cardinal markers for these regions, TH for the dopaminergic neurons, strongly expressed in the midbrain, and DARP32, the marker of the medium spiny neurons of the striatum being expressed in the striatal organoids. And in this experiment, as well as in many of the following, we have actually labeled the midbrain organoid with GFP, so we have these stem cells engineered in a way that they stably express GFP and thereby appear green. When we now take these midbrain striatum assembloids and subject them to RNA sequencing, single-cell RNA sequencing, we not only find the various types of dopaminergic neurons, for example, A9 and A10 dopaminergic neurons, so different regions of the midbrain, but we also find a robust population of the medium spiny neurons of the striatum. So we have the main cell types that are here relevant for us being present, because normally these dopaminergic neurons would project to the striatum medium spiny neurons. Now, to investigate that in further detail, we used various imaging and sensing approaches that are largely done in collaboration. So, for example, this high-resolution imaging here is done together with Ulrich Kubitschek's lab at the University of Bonn. You again, in green, see here the midbrain organoid, and we used then an antibody against TH, against the enzyme that is expressed in dopaminergic neurons, to see whether these dopaminergic neurons would actually project into the striatum. And you see on the upper left here an overview image, but then if you zoom in and do a 3D reconstruction, you can quite nicely see these dopaminergic neurons having their cell bodies in the midbrain, but then sending their axons deeply into the striatum. And then together with Peter Ertl's lab at the University of Vienna, we used sensors that would be able to measure actually levels of dopamine, because we not only want to see that they are connected, but also that the dopamine that is produced by the dopaminergic neurons in the midbrain is secreted in the striatum. And if we use these needle sensors, we can measure in the striatum pretty low levels of dopamine, as you can see here in this figure B. But if we connect this striatum to a midbrain to build this assembloid, levels of dopamine in the striatum go up significantly, indicating that indeed there's a functional connection with the secretion. To further prove functional synaptic connections, we teamed up with Rebecca Mazza's lab at the Institut Pasteur in Athens, and they used a rabies virus approach where you do a sequential infection with two viruses having a green and a red fluorescence. And just to explain that very briefly, whenever you would see red fluorescence in the midbrain organoid, you would know that there is a functional synaptic connection between striatum and midbrain, because this virus allows a retrograde labeling, and it's only transmitted in active synapses. And as you can see, particularly in the higher magnification images down here, down on the right side of the slide, you see red fluorescent cells that are even positive for the TH marker, and these are dopaminergic neurons that have functional connections to the striatum, and they have their cell bodies in the midbrain. And then to really finally see that this functional connection also goes hand in hand with the firing of action potentials, we use microfluidic devices that are developed by Regina Lutke's lab at the Technical University of Eindhoven, where we have two compartments. On the upper left here, we would have the midbrain organoid, and the compartment with the midbrain organoid is with channels connected to another compartment where we would have the striatum organoid. And at the bottom of these channels, we have electrodes spotted. So whenever an action potential is now fired from one side to another, we would see an activation of these electrodes because it's an electric signal at the end of the day. And by doing that, we get what you see here on the lower left side of the slide, namely a raster plot of neuronal activity. Together with Mario Bortolozzi's lab at the University of Padua and at VIM in Padua, we were able to not only really measure this activity, but making sense out of it in a way that we can see directionality. And the striking thing is it's nearly exclusive directionality from the midbrain to the striatum. So we see lots of firing, and this firing in really the majority of the cases goes from the midbrain to the striatum. Now, knowing that the majority of the neurons in the midbrain are actually dopaminergic neurons, we can be pretty sure that many of these signals that we see here are actually from dopaminergic neurons that innervate the striatum and there activate or signal to medium spiny neurons. Finally, to conclude this presentation, I just want to have a glimpse on the induction of aging, because as I've told you, Parkinson's disease is largely driven by aging, or aging is the major risk factor. So we wanted to also mimic this aspect of the disease in the dish, and for this we teamed up with Frank Edenhofer's lab at the University of Innsbruck, who has a strong expertise in the induction of aging by overexpression of progerin. So progerin, you probably know from the disease progeria. This induces a premature aging. So we have here now used iPS cells where we can induce the expression of progerin by doxycycline treatment, and that also leads to the expression of GFP. If we do that, we can on the one hand see that progressively from day 30 to day 60, the expression of certain aging markers increases. We here, for example, have looked at the H2AX marker, and we can very clearly see that this, upon treatment with doxycycline, increases a bit at day 30, but it particularly strongly increases at day 60. So we can progressively induce aging. And we looked at many more markers, but I just want to show you very quickly, just to stay in time, that we not only induce aging, but that this aging also goes hand in hand with the induction of senescence. This is a beta-gal staining here, and beta-gal staining indicates senescent cells. And you see that in the wild-type situation, so without progerin, we hardly see that. We see here and there a single cell, but not massively. And also in the engineered cells that are untreated, so where the progerin is not expressed, we would not see senescent cells. But in the engineered cells where we induce expression by doxycycline treatment, huge amount of senescent cell is coming up. So we can experimentally here quite nicely induce senescence. And also in an RNA sequencing approach, we very clearly see that these engineered cells expressing progerin cluster very differently, different or far away from the other cell types where we would not induce artificial aging. We find in those cells also hallmarks of Parkinson's disease. This is all summarized in the manuscript that's currently in revision, and hopefully when this webinar comes out is already public. So what I've shown you is that we can build midbrain organoids from human stem cells. They recapitulate many aspects of Parkinson's disease when they come from patient-specific cells. And we can by now also build midbrain-striatal assembloids, and in particular, we can also induce aging in these cultures. There's many more developments in that direction. We're building more complex assembloids, aiming at having an as good as possible representation of Parkinson's in the dish. And with that, I would like to thank my team at the Luxembourg Center for Systems Biomedicine at the University of Luxembourg, all collaborators. I hope I have mentioned everybody who's involved in that study, and particularly, obviously, also the funders that made this research possible. And I thank you for your attention.
Thank you, Professor Schwamborn, for this insightful presentation on modeling Parkinson's disease using organoids and assembloids. Now, let's welcome our next speaker, Dr. Rodríguez-Gatica, who will share his expertise on 3D light sheet microscopy and its advantages for organoid and assembloid imaging.
Today, I would like to present you this new technique or pipeline that was optimized in order to get most of the information of 3D samples like organoids. So first, our motivation is that, well, you know that depending the type of samples that you have, you will use a specific type of microscopy method. In general, light microscopy have the limitation of the diffraction of light that is about 250 nanometers. And if we want to overcome that limitation, then we have to go to the super resolution microscopy. Unfortunately, these methods doesn't allow to image a sample in depth that is more than 300 micrometers. So what's happening if we have a sample, a 3D sample like an organoid, right, that is between 1 millimeter and 1 centimeter in terms of size, but and we would like to see cellular or subcellular interaction, let's say synapses. Synapses are in the range of about 100 nanometers, so that fits with super resolution microscopy. But if we want to preserve, right, the complete information of the sample, then we cannot use that type of techniques. So the question is how to achieve detailed imaging of these subcellular features. So first, how to get sub-resolution features, right? When we image a point source, our microscope is introducing some distortion, and that point doesn't look like a point anymore. But so we have that the airy pattern or the deformation that is caused by the point spread function. And if we have 2 points that are too close to each other, then we are not able to resolve in classical light microscopy. So super resolutions try to sharp this PSF, right? But they have the disadvantage that you need really specific equipment, and still you will not be able to see samples that are more than 1 millimeter in size. So another way is to use expansion microscopy, where we physically separate these the elements in our sample. So how expansion works? So let's imagine that we have a sample like that, a cell, and we would like to analyze one of the features like microtubules. So first, what we normally do after fixation is to label that either with antibodies, with a direct conjugation, primary, secondary, or expressing fluorescent proteins. Then is when the expansion protocol starts, and what we do is to introduce a linker molecule that is catching the amine groups of the proteins, and in the other side will get attached to a polyacrylamide gel that we are inserting into the sample. Then in the next step, the digestion or homogenization of the sample, what we do is to try to remove everything, every element that is not attached to the gel, right, to this polyacrylamide 3D grid that we are inserting into the sample. So everything that is not attached to that is washed away, and we just have a gel with the fluorophore or part of the proteins retained. And because of this gel is hydrophilic, if we put it in water, right, we have this water dialysis, and the gel will start to grow in size. So here's an example of a mouse brain section. We can see the before and after. So after, what do we have? We have a completely transparent sample. We have a refractive index like water. That means that we don't have the needs, right, to use special objectives. We can use normal objectives. And the original protocol was made to work with samples that were about 100 micrometers in thickness and had a weak preservation of fluorescent proteins. So we went through a process of optimization in terms of permeabilization, linker, penetration of the elements, the expansion buffer, and also the digestion buffer. And now we are able to preserve fluorescent proteins, and we can work now with samples that are 20 times thicker than was reported in the original protocol. So how to image this type of samples? So, well, if you go to confocal, that is like the standard way of imaging for biological samples, what we are doing is exciting the fluorescent molecules of the sample, and then with the same objective, we are getting back, right, the excitation of those molecules. So we have to do that point by point if we want to image a complete plane. And this process is slow, right, because we have to go point by point through the complete image plane, but it's also damaging a little bit the sample in terms of photo bleaching or photo damage, because we are always illuminating the complete sample above and below the image plane that we are interested in. So one way to overcome this problem is with light chain microscopy, where we have the illumination and the acquisition in a perpendicular, right, in an orthogonal way, so like this. So we have an objective that is just dedicated to the illumination of the sample, and another one, right, acquiring. So if we generate this sheet of light, right, the objective can acquire the field of view at once without any problem. So it's much faster. So we can compare that with the epifluorescence, but with the advantage that we have really low out of focus illumination. So one of the advantages of light chain microscopy is that it's really fast compared to confocal. We have very little photo damage, almost no out of focus excitation, but we have the drawback that is really sensitive, right, to absorption and scattering of the light. Luckily, this is not a problem if we work with expansion microscopy, because as I mentioned before, we will have a sample that is completely transparent. So in the beginning, some years ago, we combined and we developed this technique, that the combination between light chain and expansion, that we name light chain fluorescent expansion microscopy, in order to take the best of both techniques, right? So because with expansion, you will have the drawback of have a large sample. So if you put that under the confocal, imaging time is increased. And you have, and by increasing the imaging time, you also have more risk to photo bleach your sample. So with light chain, we can go through big tissues, big samples quite fast, but we also have a drawback, that is that we will generate a lot of data. So that's why in the first time with our custom build setup, where we have also a revolver in order to change magnifications, we suggest that the best way is to, depending what do you want to image, go for a specific magnification of your sample. Because if we consider an expansion of 4 times, right, that we make our sample 4 times bigger, that of course will increase the resolution of our microscope by 4. So we could achieve, for example, lateral resolutions of 90 nanometers. But at the same time, if we want to image 1 cubic millimeter, we will be generating about 8 terabytes of data, right? So, and that's all in one channel. So that's why what we suggest is to have like a multi-scale approach with your samples. Now, an example, how we can analyze, right, the cellular and subcellular architecture of 3D samples like the organoids. So what do we need? So we would like to preserve the 3D architecture, especially because people is generating these nice 3D samples that try to recapitulate what is happening in vivo. So we don't want to have that nice 3D sample and then cut it into slices, because we will generate artifacts, we will lose information, and we don't want that. So we need to be able to get a whole imaging, a whole organoid mounting and imaging, and get super resolution features. So here you can see a pipeline that we report in 2022. So with a fixed brain organoids, we suggest some permeabilization but here you can use any antibody staining with conventional antibodies, and then is when the expansion, this optimized expansion protocol start playing a role. So we embed our sample in a gel, we digest in order to have a transparent sample, and then if you expand your sample in water, you will have a sample that is 4 times bigger. But you don't have to put it in water necessarily, so you can also put it in PBS, and then your sample will be only 1.5 the original size. And what is nice is that you can, even if you have a start with your sample in PBS, then you can wash it with water, expand it 4 times, and after imaging, you can wash the sample again with PBS and shrink it back. So here you can see in the upper part an example of an organoid of about a bit more than 1 millimeter in diameter, right, embedded in the gel. So you can see the original size. Then it's exactly the same gel, expanded 1.5 and 4 times. And the image is the optical section of the organoid that we had created with the light sheet. So with this, right, features that we can expand, right, our sample, our complete sample, in different with different expansion factors, plus the ability to use a light sheet with different magnification objective. We recommend to go to different scales, right? And for example, in the mesoscale, image the complete organoid if you want to have, let's say, the organoid volume, the surface area, or if you want to see, in this case, rosette distribution. In the microscale, you can see cleavage planes. You can recognize specific interneurons that are usually difficult to find if you just work with the slices, right? Because if you have a distribution of few cells inside of the whole volume, you usually have the chance that you will take a slide and you will not find nothing there. And then in the nanoscale, as I will show you, we are able to find dendritic spines and also to measure synaptic distances. What is important, right, for the people who is working with organoids is to get most of the information out of that. And as you can see, in confocal, you have the problem that when you are going deep into the sample, then you are not able to get that much of the information, even if the antibody penetrates there, right? Because your sample is not transparent, and you have a scattering of the light. So here is down is exactly the same organoid that was then mounted and expanded and imaged with the light sheet. And here we can see clearly, right, the information that was hidden when we use the raw sample and confocal. Also, structural integrity of your sample. The idea is that if we expand this sample in an isotropical way, right, that there are no deformations. So here we can see some GFP positive projections in the raw sample. Then we found exactly the same projections after the treatment. And already you can see that one of the advantages is the reduction of the background. If we compare both, here you can see that the difference is really minimal. And that is because the digestion was optimized, right, in order to eliminate, right, everything that is not attached to the gel, but at the same time is gentle enough to preserve the fluorescent proteins, because there what you can see is expression of EGFP without any antibody, and we don't want to lose, right, that information after the complete procedure. So here it's we analyze some mature neurons in cortical brain organoids. Again, here if you see the size of the sample is more than square centimeter and more than 5 millimeter in terms of thickness. So we use the mesoscale imaging just to have an overview, and then we focus in a small region of interest in order to get a volume. Here we can see now the cells And if we focus in another smaller region of interest and we go with a higher magnification, we can see that neuronal projection. And in the arrows, we mark some protrusions that, in order to properly identify if those are spines and not just an artifact of the image, we analyze that in 3D. So we are able, right, to go to that level of detail into the sample, make a segmentation, and quantify the dendritic spines. Unfortunately, this sample that was cultivated for 5 months, we couldn't find a lot of synaptic interactions. So that's why what we did at that time was to cultivate another sample for 14 months. We use pre and postsynaptic antibodies against Homer1 and synapsin. And again, we image a small region of interest, as you can see in the video, with the highest resolution. And then the idea was to recognize the GFP positive projections and use that as a mask to recognize pre or postsynaptic proteins and the counterpart. So like that, we are able to quantify either the amount of dendritic spines that we have in the surface of these neuronal projections, but also synaptic distances that are around 150 nanometer apart. And that is below, right, the diffraction limit that I mentioned in the beginning of 250. So we are able, or we have the capabilities to go for super resolution. Now we'd like to show you an example, a project of Dr. Kyriaki Varmpa from the lab of Professor Schwamborn, where they produce assembloids in order to get a better model for the nigrostriatal pathway. So they use midbrain and striatum organoids that they fuse in order to recreate this connection. And these samples have something special, is that they are embedded in like a matrix gel that is called Geltrex. And again, what they would like to have is the 3D preservation of the architecture, be able to image the complete sample, and in the same imaging session, be able to choose a specific region of interest and see if there is projections of thyroxine hydroxylase positive cells from the midbrain to the striatum assembly. So this is one of the samples. So you can see in blue the nuclear staining, the GFP positive cell corresponding to the midbrain organoid. So you can see that it's smaller because was cultivated only 25 days. And we also use TH to see the connections, right? Even if the sample here looks smaller, it's just because in the surrounding there is this Geltrex that was also preserved in the beginning. But thanks to the light sheet imaging, and this time we use the ultra microscope blades from Miltenyi with the 4x magnification, we are able to scan, go through the sample and see, right, the interaction between these two organoids that are forming now this assembloid. Now that we know that we have the information, right, of the complete sample, we would like to go to a specific region and see, right, if there is interaction between these two organoids. So without taking out the sample, right, we just change to a different scale. Then we focus in this small region of interest where we would like to see the interaction between the midbrain organoid and the striatum organoid. And for that, we use a 25x objective that is mounted in our Blaze microscope. And here we can get a better resolution, right? So we can see clearly, for example, the nuclear staining, and the projections of these Th positive cells that are going from the midbrain to the striatum organoid. And because of the better quality of the image, right, we are able to do a segmentation, and we should be able to quantify and to validate this nigrostriatal pathway connectivity. Of course, we did tests with more or different organoids, and here in the video you can also see how the projections of these GFP positive and Th positive cells are going from one organoid to the other inside of this assembloid sample. So we can identify these neurons that, if you think that you have a sparse level or a few quantity of this or amount of these cells, is really important to keep and to be able to scan the complete sample without cutting. First, because neurons could project in any direction. So if you decide to cut your sample to analyze this, you have a high chance to cut these projections and lose the information of where it's going. And if you have few cells, then also if you are able to scan through the complete sample and then magnify and get a proper resolution, right, then also you increase the chances of finding these neurons into the sample. So as a summary, what I would like to show you is that we have a method that will work with really extended samples, about 1 square centimeter, more than 3 millimeter in thickness. We are able to have super resolution features. What is important is that we can preserve autofluorescent proteins, GFP, M-cherry, TD tomato, M-cerulean, and so on, and it's compatible with commercial antibodies. So if you have a sample, right, that you have to that you already know how to prepare, that you already have the antibodies, then you don't have to change nothing, just to apply this optimized protocol and bring it to the light sheet in order to have a nice data. And what is important is that with this, right, methodology, we can image our sample over 5 orders of magnitude, right, without losing any information. So that's probably the most important point that I would like you keep in mind. So you can go to different scales during the same imaging session. And with this, I would like to say thank you to my work group, the previous one where I was in the biophysical chemistry group, and my new work group in the neuroconnectomics with Professor Martin Schwartz in the Uniclinicum here in Bonn. So thank you, and ready for questions.
Thank you very much all for those beautiful presentations. We already have some questions coming in from the audience, so we can get right to those. So first question is somebody who is a little bit less familiar with the topic, and they're asking a question: do these organoids also mimic immune response to the drugs that are used for Parkinson's disease? Is anybody from the panel able to address that question?
Yeah, I guess that goes to me. So midbrain organoids in their normal fashion, in their kind of first generation, would not have the immune system because they are derived from neuroectodermal stem cells. So from the ectoderm, the immune system comes from the mesoderm, so we would have no immune system in there. However, I've showed you the second generation that also contains microglia. So we would derive them separately from iPS and then simply mix them in. And these indeed mimic immune functions, and also these microglia in there would, for example, respond to drug treatment, yes.
Thank you. And the next question is, as the organoid models become increasingly sophisticated, What are the ethical implications of using human-derived tissues for research purposes, and how can we ensure that the models are used responsibly and ethically?
Guess that goes again to me. So on the one hand, everything is derived human stem cell based, right? So there is a first line of defense, if you will. So we have to have obviously ethics approvals to work with these human stem cells. And I can only talk about the national regulations we have here in Luxembourg. So I have to present my project in front of an ethics board and answer questions and get approval. But then next step is obviously for organoids, which are complex. And then there's this whole idea that they develop a level of self-awareness and these kind of things, which I would say we are pretty far away from that. Nevertheless, this needs to be ethically controlled. I think it has to be overseen by an independent board. And I think the procedures, at least what I can say for our situation here, are in place for that.
Thank you. And the next question is for Juan. Regarding the sample preparation, can you elaborate on the digestion process that makes the sample transparent?
Yes, sure. So the digestion buffer is basically made to work with proteinase K. So we have EDTA, 1-edine, tris, and triton. And the idea is to use proteinase K to break all the protein-to-protein interactions or protein-to-lipid interactions. So everything that is not attached to the gel, right, by the linker molecule will be washed away. So that's why finally the sample is transparent, because we have basically all the lipids gone. So that's how we make the digestion, and we try to always optimize to the use of proteinase K. So proteinase K is usually optimal at temperatures of 50, 55, and a pH of 8. So we don't we optimize the pH to that number, but we don't do a digestion in a high temperature, because that will degrade other proteins, like for example fluorescent proteins. So we decrease the temperature to 37 degrees. So it's a mild digestion that maybe it will take a bit more long to get the sample transparent, but at the end you will be able to preserve more information and to destroy less of the proteins that also you would like to preserve.
Thank you. And the next question is, can 3D imaging be used to observe mouse brain regions as well?
Yes.
Yes.
So I mean, well, the blade, with the blades, there are also other techniques, not only expansion, that you can use to clear the tissue. And depending on that, right, you can choose also between different objectives to match the different refractive index that these clearing methods could have. Like you have the organic solvents that usually have like a higher refractive index, but in the blades you also are able to exchange not necessarily the complete objective, but just the cap in order to match that those requirements. So yeah, I mean, it's really versatile tool if you want to put a transparent sample under the light chip.
So it's even possible to image a whole mouse. So not only a brain or an organoid, but even a mouse.
Thank you. So Jens, you touched on this a little bit. Maybe you can address this as well. Somebody's asking, you mentioned that the organoid recapitulates most of the midbrain cells. The question is, does the organoid also possess microglia?
Yeah, that pretty much is in line with my first answer, right? So an organoid or a midbrain organoid that derived from new epithelial stem cell does not contain microglia. It comes from the neuroectodermal lineage. But you can mix in microglia later that you separately derive from iPSCs. And that's by now actually the standard model we are using most of the time. So yes, you would have microglia in there.
Thank you. And the next question is, can the 3D imaging be used to... Oh, no, we answered this one already. Next one is, in the expansion protocol, did you test other types of endogenous fluorophores other than EGFP? Whether these are preserved and whether it was preserved as well?
Yes. So yes, I test different endogenous fluorophores, right? Like EYFP, TD tomato, M-cherry, M-cerulean, and I think that most of them are working fine. Usually when we have the problem that if there is just few amine groups or no amine groups in that fluorescent molecule, that will not be preserved. But it's possible to fix that if you change the linker molecule, right, in order to catch one of the one group that is present in this fluorescent molecule. That's, I would say, the only restriction. Also, then when you preserve this, for example, fluorescent molecule, and after the digestion, if you notice that the intensity is not that high, you still are able then to do a post-digestion labeling, antibody labeling, because you will have, right, the signal of the fluorescent protein that it's a bit weak, but that means that the epitopes are available, so you can use like a primary or secondary in order to boost that signal again.
Thank you. The next question is for Jens. There are some reports that have found some relationship between the pathological conditions and mechanical properties of brain tissue. Question is, have you explored anything in regards to this to better mimic the human pathological conditions in your organoids?
Yeah, that's a very interesting point. We have not touched on that. So what we know is that in order to grow brain organoids, we need relatively soft matrices like gel tracks or matrigel. And if you get more stiff matrices, that would not work. But we haven't done anything in the direction of pathology, although I fully agree it's interesting, but I cannot really answer anything on that.
Thank you. And the next question is, how can patient-derived iPSCs be used to generate personalized organoid models that accurately reflect the genetic and phenotypic heterogeneity of Parkinson's disease?
Well, a priori, because it is iPS-derived, it is patient-specific. So each model that we produce is, by definition, personalized.
Wonderful. And another question, which is a bit more technical, can you please clarify whether you're using the 4 times objective for collection of fluorescence? In this case, is lateral resolution very low?
Well, we use the 4x just to have like an overview of the organoids. And yes, the resolution will be low with the 4x, but you have to consider that the resolution is always, it's depending what do you want to see, right? If we want to see a complete body, right, the complete organoid that is like almost like more than 1 millimeter in size, then the resolution of the 4x is good enough just to see the complete, let's say, element, right? Now, when we show these high resolution images, what we did was to change to a higher magnification objective. For the assembloids was a 25x with a high numerical aperture, so in order to have better resolution images. So maybe he was confused because I was talking about 4 times or four times expansion. But that's one thing. So if you expand 4 times, then you improve the resolution of your microscope by 4. And that's different from the objective that you use. So just to make clear, so we use low resolution objectives to have like an overview or to measure things in the mesoscale, and then we can change to different magnifications to go to the nanoscale.
Thank you. And another question we have is, how long does it take, how long is the process from sample expansion for the imaging? So how long does it take to process?
Well, the sample preparation for expansion depends on how big is the sample, because what is important is that the linker molecule, right, penetrates the complete sample. Also, this polyacrylamide gel matrix, right, that is responsible for the mechanical expansion. So, for example, a sample, imagine that it's a sample of like a sphere of 1 millimeter. Usually you have that sample, the complete process will be like 3 days, more or less, to have a completely transparent sample that will be ready to be imaged under the light sheet. now the imaging on the light sheet is like, I would say, not more than 1 day, because it's really fast. And I mean, 1 day if you want to really go with several regions of interest and check with different magnifications.
Thank you. Okay, we have time for one more question. And the question is, how can we leverage advanced 3D imaging techniques to bridge the gap between in vitro organoid models and in vivo human brain imaging to provide a more comprehensive understanding of the spatiotemporal dynamics of neurodegeneration and Parkinson's disease? And I guess a follow-up to that is also to enable the development of new therapies.
Juan, that goes to you.
It's well, I think that all this. So if we want to mimic, right, what is happening in 3D, I mean, this 3D imaging is completely necessary. I mean, it's already easy to see how much like a model, right, is changing, right, from a 2D culture to these organoids. So I think that is. I think that the question is gone. I think that is really important, especially to be able to get the complete 3D information without altering the tissue. I think that is actually the most important part. Independent the method that you use to make your sample transparent to image, you need to always think in the integrity of the sample. Right? So I show you in one slide why we are using this technique is because we test enough and we are sure that we are preserving, right, the 3D morphology of the sample. If you are able to get that, then you can start to translate that and maybe at some point, right, bridge the knowledge between these tiny models to what is happening in a human brain. Also making the link with, for example, MRI imaging, like functional imaging, and try to merge all this information.
Fantastic. Thank you so much. Thank you to all of you for that discussion and for your wonderful presentations. We appreciate you joining us here today. Thank you. If anyone has any further questions, please consider reaching out to the speakers directly. Their contact information is shown on the screen. As a reminder, the webinar will be archived on the Scientist website. You'll receive an email notifying you when the on-demand webinar is available. Make sure to follow the Scientist on social media for updates on future webinars. I'd like to thank everyone who took the time to join us today, and especially those of you who shared your questions and comments. On behalf of the Scientists, I'd also like to thank our speakers again, Anisha Munoz-Bull, Jens Christian Schwamborn, and Juan Eduardo Rodríguez-Gatica, as well as our sponsor, Miltenyi Biotec. Thank you, everyone.